Hello and welcome. I'm Claudia Rizzini. I am the Executive Director of the Radcliffe Fellowship Program at Harvard University. It is my pleasure to introduce today's talk by this year's Grass Fellow, Bruno Correia. Bruno is a biochemist fascinated with proteins, their structure and function. For several years now, Bruno's laboratory at the École Polytechnique Fédérale de Lausanne in Switzerland has been working on the design and adaptation capabilities of proteins. One of his specialties is to combine experimental and computational techniques to manipulate the structure of proteins and therefore their function, primarily in the area of immunology and synthetic biology. His group is working on one hand on repurposing proteins already existing in nature. On the other, they are designing novel functional proteins. The goal is to use these novel technologies to design new vaccines, find new cancer therapies, and much more. For example, in 2014, while working with RSV, a respiratory infection affecting primarily children, he demonstrated for the first time that computer-designed proteins could result in a functional antibody. Bruno has been lauded by his colleagues for his constant willingness to innovate and collaborate with other researchers. He has made his research protocols available to the scientific community and received several awards for excellence in research and teaching. Bruno's dream, however, was to be the front man of a rock and roll band. But he had to settle for being a scientist instead. Don't call him a professor, though. His view is that titles don't put people at ease. I'm so happy to welcome Bruno Correia.
It's going to be a little bit of an unusual talk for the typical or the more typical themes that we have here at our Radcliffe series. But today I'll tell you about proteins and the work that our laboratory does with them. And so just to introduce you to what the menu it's going to be. So I'll start relatively broad in order to tell you more about what this class of molecules actually are. And so what is really the challenge on how to create these novel proteins and why do we actually need to do that? Then I will follow through very smoothly on how to use these proteins to modulate activities in the immune system. And I will finish up with how do we actually represent these molecules to the eyes of a computer so that, in fact, we can manipulate them in the way that we're interested. But I will try not to be too serious about it and rather just give you overall messages so that you can take a few things home that you can think about. And I'll start with a citation from one of our fellows, where it's written in one of the books that the fellow wrote, "Biology is goofy." And it is an amazing, simplistic, and accurate way of describing biology. And the reason why, in fact, it's goofy, it's not really because it's goofy, but it's rather because it's complex, right? And there's a lot of behaviors which we would like to predict much better in biology, but we just can't. And the reason why we can't is because, again, it's inherent complexity. And the conversation is, if you want to think about the basic unit of biology, you can think of it as a cell, right? Of course, this depends on the level of resolution that you want to think, but a cell is the basic living unit. And so in that sense, if you want to think about a cell, you can actually kind of like point out two different things in a cell, right? So it has the nucleus, it has the cell membrane, so DNA, organelles. But I think more important for us to think here today is to think that, in fact, what cells are, they're just kind of like biocomputers. They take inputs and they spit out outputs. And that's basically what they do. They do it through very complicated mechanisms, but in a first approximation, that's what's happening. Now, if you want a real picture of a cell, it's more like what it looks, right? So under a microscope, that's basically, so you start seeing all these kinds of structures that are here schematically represented. Now, in order to have these biocomputing units to work, you need molecules, right? And if you want to think about what basically some of the major classes of the molecules of life, you can think about DNA, where our genetic information is stored and it's passed from generations to generations, to lipids, to glycans. Oh, so let's see if I can get this. Okay, here we go. To lipids, to glycans, and to proteins, right? So, and as you can already guess, our main subject of today, it's going to be proteins, right? And so proteins, right? So if you also want to think about them in a figurative way, you can think about them as the molecules that mostly do the job inside cells. So all the functions, or many of the functions that occur in living systems, are carried out for proteins. And so how do we actually get these proteins? And the idea of these proteins is that you do have the information on how to make them stored in your genetic information, right? And that's what you see. This mouse keeps playing around with me. There we go. That's what you see on this side of the slide, and then ultimately this information gets translated into amino acid sequences, and these amino acid sequences then adopt a three-dimensional structure. Now, the important thing about this three-dimensional structure is that it lives in the nano world, so you can't see it, right? It's not like, so in a way, right, when you tell somebody that you're working with molecules, sometimes you kind of have to believe what you're working on, because it's not part of the range of sizes that we can see, right? So these little structures, they live in the nano world, so they're very, very tiny. However, we do have techniques in order to understand what their structure looks like, and as it turns out, proteins are in fact very cute, and I mean they're very, very cute. So thinking about the different kinds of things that they can do, ultimately, you can have these things that are called antibodies. I think you guys are all very familiar with antibodies by this point. And it's actually, we're going to talk a lot about it throughout this talk. But this is, if you didn't know how it looks, this is how it looks. Basically, this is like the structure of an antibody. They can also be serving purposes for storage of different chemicals. They also do transport functions. They also do communication functions in cells. They are also important for maintaining structural properties of our tissues, and also to perform chemistry. That's very interesting, right? So you have like a protein which basically takes one molecule, as you see there, the maltose substrate, and produces a different molecule. So basically chemistry happening inside of these molecules. Now, how do we think about these proteins and basically what we call the dogma or the paradigm in molecular biology? So the dogma goes this way, right? So I haven't told you much about these sequences, but I will tell you in the next slide. So you start with a sequence of a protein that you can write, and then this sequence adopts a particular structure, and this structure translates into a particular function when it's inside cells. Now, sequences. When I showed this the first time to somebody that it was not really from my field while I was here, so people, but are these real words? No, they're not real words. They're not real letters. They're just symbols. But if you want to make a figurative comparison to the words you write in books, yes, you can think about this as the words that nature uses to write a text, a function in your cells. But in real life, they're not real words. They actually have chemical structures, so they're just symbols. And so each one of these letters has a different chemical structure, which I'm showing you here on the right. And what's important to know about these chemical structures, you can spare the details, but some of these chemical structures, they have different properties. So some of these chemical structures, like water, some of them don't. And then this is basically what causes these molecules to adopt different structures. So, and that's what I'm going to try to show you here. Let's see. So now, since you have one of these sequences being made in your cells, so you can think about this as just a bunch of different letters together. And then when basically this actually occurs in your cell, the process that we go through is that one that we call protein folding. So basically, a protein gets synthesized, but then because you have some of these letters that like water, some of them that don't like water, they basically sort of like clump together in order to basically obey their basic chemistry properties. And that's ultimately how you obtain these beautiful structures that I showed you. And this is ultimately, if you think about it, it's basically the most basic need for a protein to be in a cell is that it needs to be folded and hence being functional. Okay, so that's good. However, and if we actually study many different organisms that are out there, nature has in fact provided us many different proteins. And of course, these proteins are brought up to us through processes like evolution. So we evolve, and our proteins evolve with us. And basically, this is how we change the traits of different organisms. But now, if you're a protein engineer, you have a little problem. And that problem is a problem of size. And so if you look at these numbers that I have here on the left, so let's say that I'm an engineer, and now I want to play nature, and I want to make these new proteins in the computer, right? And or by hand. So if I have a protein that is made out of 50 building blocks, so 50 amino acids, these 50 letters that I told you about, I basically have potentially 10 to the 65 different combinations. I know that it's hard for you to visualize this number. It's also hard for me. But it's a really big number. So it is not impossible. It's not possible for us to try and explore each one of these proteins, right? And so just to give you an idea of the number of the atoms in the universe, well, that's that. So if you want to do a protein of 100 amino acids and you want to try all the possible combinations, you're already far beyond it by many orders of magnitude. So this is just simply not possible. We cannot create these proteins just at random, because the likelihood to hit something that actually it's viable, it just doesn't exist. Now, what we also realized as we start sequencing more and more organisms out there in nature is that just due to the sheer amount of space or amount of possibilities where we can create new proteins, that nature has not explored all the possible proteins that exist, which basically leaves, and so, and I like to represent this as being the potential protein space that exists, and really nature only sort of like poked in a few spaces, and sort of like, and it didn't need much more than that. We're all happy, we're all healthy, life is great, we don't need to sample much more than this. But now, as it turns out, modern societies, they have a lot of needs. And we keep coming up with more needs. We need new vaccines, we need new antibodies, we need new enzymes to get rid of plastic. So there's a lot of needs where nature can happen. And that's why there's this effort to try to explore the different space of proteins that nature has not made. And of course, this at some point took place, this exchange, and this is a real conversation, by the way, where my neighbor actually asked me, so, well, why don't you just make them fold? And I'm like, well, I wish I could, and I've been working on it for like 15 years, but it's complicated. And so it's very hard to actually figure out one of these sequences, one of these words, where you go to the lab, you express this protein, and you see how it works. So let me tell you now about how we do this and why this is important, right? So again, this talk is about engineering new proteins that nature has not created. And to this exercise, we call protein design. And so what's protein design, right? So protein design, in the end, is the opposite of the first scheme that I showed you, where you basically define which function you want to have for the protein you want to make. And then, well, if you are a person like me, you like to think about it in three-dimensional structure. So you create a new structure. And then this structure will have to translate into some sequence. And then once you have this sequence, then you can go to the lab and test it. And I'll tell you how we do this. So, and of course that, again, I was telling you, and to give you one more comparison on how we actually do this, and what you see here is essentially perhaps one of the first molecular modeling exercises when Watson and Crick, in fact, figure out how the structure of DNA actually came together. And so, but they were doing this by hand, right? So they were putting the atoms three-dimensionally by hand, which is something that we don't need to do today, thank God. But basically what Crick said by that time was a very interesting thing that it took to me for a long time, is that if you want to understand function, you need to study structure, which of course the corollary of that is if you want to engineer function, you need to modify structure. And that's all that I'm going to tell you. And so if you want to think about this in a very easy way, you can think about this idea of protein design as sculpting, but you're just sculpting at the nano world in a very microscopic range. So you don't get to see exactly what your sculpture look like. But let me give you a bit more of, and I already alluded to this, but now I just want to show you how we actually do this in the computer in a very high level way, of course. But essentially what the exercise boils down to is this idea that you have this yellow structure that you see here, but now you want to find which letters, or which amino acids in a more technical term, are going to be able to make this structure to exist. And so, and the computer, that's all that it's doing. Of course, you can use many different kinds of algorithms to do this, but ultimately what these algorithms are trying to make you, to help you with is to search through this large space of combinations and tell you which ones are the words that are actually going to work. Very good. So now, even to give you a better perspective on how we do this in a daily basis. So, and again, I told you that there's many different applications. But again, so now that you can picture how the process goes in your mind. So, you have like some guy, in the middle of a lab typing in a computer. He runs some simulation. That computer sort of like produces an output, which is one of these protein sequences. And then what happens, right? And what happens is very, very interesting, at least to my end. And the reason why it's interesting is, so let's say you have this protein sequence that you created in the computer. You type it into a web server, which basically translates it into DNA. You order this DNA from some company around the world. You receive this DNA in your lab. You put this DNA into cells. And basically, the cells now are going to work as being your little factories to make the proteins that nature has not done, but that you did in the computer. And, and then it goes through a process. You grow a lot of these cells, and you shake, and you lyse these cells, and now suddenly your protein sort of like opened up, and suddenly you have this protein that nature has not made, the computer has made for you, and now you can work with it in the lab. So that's to give you our daily bread. That's how it works most of the days for us in the lab. Now, I will switch here towards more the applications and some of the work that we've been doing. And so, did I mention that vaccines work? So I'll take sort of like a little bit of a brackets here. So yeah, we do live in times where facts and data are challenged in a way which is absolutely scary, and that anybody that can write 200 characters somehow can discredit lives and works of people that dedicated their life to science with no interest. And I think that the vaccines are one of those interesting aspects where really you have evidence, and I'll tell you more about what the evidence looks like. And suddenly 200 characters put everything at stake. And, it's, to say the least, a little perverse and very concerning for a society that wants to evolve, I would say. So now, vaccines work, right? So let me just show you this slide and mostly why they work, right? And the reason why they work is because there's no wrong way on how to plot this data, right? So before vaccines, you had millions of people dying, right? Or you have millions of cases of infections. After vaccines, in many cases, you put them down to zero. Not in every case, but in many cases, you put them down to zero. So for me, it really doesn't matter how you plot this data. If the vaccines are good, they really work very well. Now, we do have a problem, again, in modern societies, is that we're still missing a lot of the vaccines that we need. And here I'm giving you some examples. And to tell you more about the work that we've been doing in vaccines, I'll give you a crash course on the immune system. And basically, very, very little. Our immune system is perhaps one of the most fascinating organs that we have in our body. I'll also tell you a bit more about it in the next slide. But we will focus on the adaptive aspect of the immune system, where we have humoral and cell-mediated responses. Humoral responses, they will make antibodies. Cell-mediated responses, they will make T cells. So for now, we leave it at this, and throughout the talk, I will tell you more about what antibodies are and how can we actually modulate these T cells to control the activity of the immune system. So I already told you a bit about antibodies, but ultimately you can see here how the immune system is in fact located across our body. So it's not one of these organs that it's only located in one side. So it's some people actually say that it can be as complex, perhaps even as the brain. So very interesting system if you want to learn more about it. And today we focus on this, or at least at this part of the talk, we focus on B cells and antibodies and what are the problems that we're facing in antibodies. So the reason why we're all here and healthy and we can live sort of like very great lives through many, many years is because our immune system has incredible capabilities. And part of those incredible capabilities is that it basically can make molecules at will. So you see a new virus or a new pathogen or something that it has never seen before. And it's basically able to make molecules to neutralize that virus, right? And part of the reason why it can do this is because it can make the estimated numbers, nobody really knows how many molecules it can make, but it can go to 10 to the 18th, again, an enormous number, okay? So, and these antibodies, they can, just to tell you how this actually works, so your immune system sees one of these proteins, so here is a representation of a protein, and basically, once it sees and it has never seen it before or it thinks that it's foreigner, it basically attacks it with this antibody. So antibodies now are sort of like binding, attaching to it, and just making sure that it gets cleared from your body. Now, one of the problems that you have, for instance, for vaccine development is that oftentimes you do have a good target for your immune system to be triggered, but the immune system, for some reason, triggers to a place where you don't want it to be. So part of that antigen, which is not so useful for your defense, and rather than actually triggering antibody responses towards this part of the molecule, what you actually wanted would be to trigger to this part. And of course, that this idea is, well, look, if I can make better antibodies, I can be much better defended than if I make bad antibodies. So to speed up the thing a little bit here, the virus that we worked out through several years now is the respiratory syncytial virus, which, funny enough, when I started working about it, the reaction of people was, which virus? But a lot more people know about it now, which is very interesting, which is not a good thing, but it just came up again because of all these constraints that we have had in the pandemics. And so this is respiratory syncytial virus. And what we're going to do here is to basically learn from natural immunity. And so to basically give you a very simplified version of this slide, so we're going to look into what actually the human body does in order to neutralize the virus, so which type of antibodies it actually creates. And then we leverage that information to engineer novel immunogens and use that for vaccination again. So, and the process on how this works in the computer, and of course these proteins are not really robotic, but this is just there for so that I could also have some robots. But the way it works is, you make these simulations where you start with this green protein, which is the viral protein, and you have a part of this protein which is identified by antibodies. So you take it out, and essentially you start simulating all this process in the computer by building these structures. And what you see there is it's exactly what happens in the computer. Once you settle in a structure that you're happy with, then you start folding this protein around this structure and trying to find which sequence now can make me this protein. That's that, right? So that's what's happening there. Now, in that sense, just summarizing, you studies from many people and also over several years. What we did this with RSV. We took several pieces of this virus, created new proteins around it, and then tested it in animal studies to see which kind of antibodies they actually would elicit. And in fact, they did elicit neutralizing antibodies. So that's part of the work that we've been doing in the lab in order to try to attract the attention of the immune system to the right pieces of structure that we deem that are better to have a good immune response. Now, changing subjects. So I told you that cells are biocomputing units. Now, one of the dreams that we had was really, well, can we now, so if cells are really this small biocomputers, can we actually make new hardware for them? Can we create proteins that can modulate the behavior of these cells? And so you see here this more or less in a very schematic way where you have these proteins that are just schematically represented in terms of the kind of components we could engineer. And so, and one of our interests was also, well, currently cells, engineered cells, are actually being used for human therapies. And these therapies are called CAR T cells. So for those of you that have never heard about this oftentimes sounds like science fiction, but it's actually how sophisticated technology has become. And so, and I'll just spend some time to tell you exactly how this works, right? So let's say that, often this treatment currently is used mostly on oncological patients and for very specific types of tumors. But what happens here in a very summarized way is you have a patient, and a specific type of cells, which are T cells of these patients, are collected. Once they are collected, you expand it outside of the body of the patient. You engineer it with a new protein, and then these cells are infused back again into the patients to be able to go and eliminate your tumor. So I know that it sounds like science fiction, but this is current medicine. It's not cheap and it's not scalable, but it's saving people's lives at this point. So you can obviously think that this is a very different type of drug than the one we use, which typically is a small molecule or an antibody. This is a living cell. And of course, with great power, there are great limitations that also come, right? And so clearly, when we started engineering, when the community started engineering these cells, there was sort of like concerns about safety, right? So like, well, when you infuse these cells back, what's going to happen, right? They might just go crazy. And they did go crazy. And this is what you're seeing here, that there were some cases where clearly things didn't go right. But that's obviously research and medicine, so things happen this way. But what we saw here was, well, can we actually now control this? Can we actually now create these molecular components to control the activity of these T cells? And so how did we do this, right? So I told you that these T cells, ultimately, they get engineered with a synthetic molecule. And so with a synthetic protein. And so basically what this protein does, in this region, it recognizes the tumor. In that region, it turns on the T cell. And so basically when these two things happen, this basically is these cells are able to eliminate tumor cells. So what we thought that we could do was, well, let's maybe create two proteins that in case I give a small molecule drug to these cells, that I can basically eliminate the activity of these T cells and basically have a safety switch. So basically like a light switch, right? If I turn on the light, things are working. If I turn off the light, things are not working. And this, of course, would be something that you could use in living people because you can actually use these compounds and you just take it as a pill. Right, so, and we tested this principle, and ultimately I won't drive you too much through the data, but rather just to tell you that in fact, it kind of worked. So we could actually infuse these cells into mouse models and then test, in the presence of the drug, are these T cells active or are they not active? Very good. So as the last part of my talk, and I really only have a few minutes left, I would like to tell you about protein representations. So oftentimes, we tend to think about how do we actually represent proteins to the eyes of a computer so that we can extract the maximum information out of these structures. And ultimately, perhaps the most traditional representation of a protein is this, where you draw each atom that corresponds to one of these amino acids, and essentially that's how the object looks like when we model it in the computer. There's of course other types of representations. You'll see more sort of like a high-level representation, and then there's one representation where we started working. Well, what if rather than working with proteins with individual atoms, we actually just represent them as shapes? So ultimately, when we have shapes, we can in fact map and learn different things about these molecules. And again, you don't need to pay too much attention. I will make a parallel for you, which I think it's much more important. So let's say that, as you know, these days, there are many computational algorithms which can do image analysis and basically tell you things that are absolutely non-obvious, right? Are you sad? Are you happy? Where do you come from? So the parallel here was basically the same kind of idea. Can I find patterns in these structures which basically tell me about what do these molecules do in nature? And the answer was, well, yes, we can do it. And so just to give you an idea on the algorithm, but we won't spend too much time here, we'll rather go to the applications. We can map into these proteins different types of properties, both geometric and chemical, and we can learn things about this. Now our idea was, can we actually use this type of framework to create new molecules? So, and here, just to show you very quickly how we actually did this, one of the advantages of this technique was that it was extremely fast. So what we could do was, in fact, let's say that I want to create a protein to bind another protein, and then I'll tell you what the application is. But the idea here is, so how can I do this, right? So because the type of computation that we were doing was so fast, we could actually screen a large number of fragments. So we could do sort of like, millions and millions of fragments very quickly and try to understand, can I find a piece of a different protein that binds to mine? And in a way, kind of do the same thing that antibodies do. So if I bind to something, then I might be able to neutralize it and just eliminate that activity. And that's ultimately what we did here with, again, our good old friend SARS-CoV. So we take the spike protein, so this is the protein at the surface of the virus. We try to find a molecule that can bind to it. We test we make all these calculations in the computer, test it in the lab, and then you can just look at this guy right here and forget about all the rest, where eventually you end up creating molecules that are able to neutralize the activity of the virus. And so, of course, we thought that this was pretty exciting to us by trying to use this type of approaches. So I'm very close to the end, and so I just hope that you can actually agree with me in this case. But if you don't agree, that's also sometimes even better. The limits for this type of research are really our imagination, and there's many things that computation can do to help us, essentially to explore this unexplored space, to create different types of molecules that can make our life better and hopefully make also the life of our planet better. And one of the things that I've been very excited to do here while I was in Radcliffe was really to think about how can we actually control what our immune system reacts to. It's clearly, one of these sort of like overwhelming problems which I never even dared to think before, because I think there's just so many aspects of complexity about the immune system that it's almost pretentious to think that you can do this. But I think it's maybe not pretentious to at least try. And the idea here is to try to understand the features of these molecules and their structures that make it more likely to the immune system to react to it. And the types of implications that this can have is basically this idea of creating vaccines that work better, or also creating new protein-based drugs, which have a higher amplitude of applications, which we currently can't do. So, the last and most important slide. One thing we should clarify right now. Science is a team sport. Nobody does anything alone. And I doubt that really there's many out there single people that can do very big transformations. So none of this work would have been possible with the team that I've had now in Lausanne for the last 8 years, which is mostly made out of PhD students and some postdocs. And really, I think we've been trying to tackle hard problems, and I always tell them, well, most of the times things don't work, but when it works, it's really amazing. And I've been really very fortunate in being able to share these times with them. and also with a number of collaborators which also bring aspects into research which is impossible to have on your own. So with this, I thank you for your attention, and I be happy to take any questions you might have. All right. 36 minutes, huh?
Yeah, doing well. Okay, so there are questions, but I'm sure we'll have time for a few more if you want to ask them. So let's start. How can you determine if the new proteins you create are not dangerous to humans or other life forms?
Well, good question. You know, it's very hard to always give absolute answers for problems you don't know the boundaries. So it is hard to say that a protein that you make might not do something completely crazy, but it's very unlikely. And also, the reasons why that is because oftentimes when we work in these molecules, we have some type of idea of what they are already supposed to do. And also when we test them in super controlled environments, right? Mm-hmm. So these are not entities that can somehow replicate on their own and sort of like, you know, spread through the world. Oftentimes we make them once. The first thing that we always check is, you know, are they toxic to the cells that they grow? And if they're toxic to the cells that grow, well, you know, we won't go there. But that's generally a very small aspect of the complexity of what your question entails, clearly.
Following up, I guess, on what you just said, the second question is, can you say more about the unintended consequences of engineering proteins and the ethics and regulation of the field?
Yeah, so in general, I should say that when it's about just to engineer proteins, oftentimes you don't have a lot of limitations. And again, these molecular entities, they have sort of like a very limited scope. It is true that when you start engineering on the next level up of cells or viruses, things change. And I think they do change. And also when they change, I think the security and the safety concerns that we have and measures that are taken, they also change. So I don't necessarily, so here's the deal. I think that a lot of bad things can happen, clearly. But I think that the amount of good things that can happen is infinitely better than the amount of bad things that can happen. Does that make sense?
Yes. Although
that's not the answer you want to hear.
Are taken care of. Okay.
and there are, you know, of course, you know, there are ethics concerns and there are certainly ethics checks. It's not like we're out there on the Wild West, you know, doing things without having precautions. Clearly.
Yep. There is a question about CAR T cells treatments and autoimmune disease. Can they be used for autoimmune disease in addition to cancer?
Yeah, there are some evidences in the field that this type of therapies can actually be used for other issues that are not cancer. I think if you think about it, you know, in a way, that's probably how our future is going to look like, right? I guess for many conditions where you do need biological entities that are more powerful than single molecules, we will be delivering a lot of therapies in cell-based formats. So the answer is broadly yes. Now, of course, one has to look into the details.
Yeah, and probably a lot more work needs to be done on that.
Oh, yeah.
Let's see the next questions. Let's go to this one. Can you please speak more about the phenomenon of vaccine hesitancy and how your work and the way you make it comprehensible may be able to shape public opinion?
Yeah, I wish I could do more of that. I have to say that I'm absolutely a non-expert. And I come from a very biased place because even culturally, where I was brought up, vaccines were compulsory, nobody questioned. That was it. That was the conversation. Start and end of the conversation was there. So I'm very biased because, you know, I test vaccines in my lab and I see the results every day, and well, when they're good, they really work. So I think the issue here is when opinions or beliefs try to overcome facts and hard data. And I think the only way that we can somehow try to address this vaccine hesitance is by talking to people, and also again trying to meet them where they are, and try to tell them where, so like, well, look, here's the evidence for why it works. And I think that's as much as we can offer. But I think we cannot stay on this place where, well, no, look, it just works. That's not the way it's going to go. We will have to somehow walk a different path, and again, based on data, evidence, facts, show to people that their hesitancies might not have many reasons to happen. I think that's the idea.
Let's go into the weeds of what you're doing. What statistical challenges do you see in your computational designs problems?
Very good. So this also takes us to these new domains of machine learning approaches and what people commonly call artificial intelligence, which works very well for many, many domains. And it works particularly well for domains where there's very large amounts of data. We face the challenge that although for us it's a lot of data, for machine learning algorithms is sparse data. And as such, they do have issues learning the things that we want them to learn because our data sets are not big enough. So there is statistical challenges in this sense that oftentimes for the most advanced technologies you have to play tricks in order to make sure that that you can leverage the best out of them.
The next question, let's see, are there other ways to engineer cells outside of computational designs? Of course, here we're engineering proteins, not cells,
yes.
right?
But it's okay, it's kind of the same. Yeah,
okay.
Yeah,
if so, what are the advantages of these other ways?
Well, so there are different ways of engineering cells. I'm sure you guys have all heard about genome editing, CRISPR, Cas9, so all these kinds of proteins that have been discovered and have been awarded Nobel Prizes that have the capability to go and change one's genome. So there are different technologies to do so. You don't necessarily have to use proteins designed by computers. You can also engineer proteins in different ways. Now, I think what is more interesting and that I would like to think more as more like a futuristic, I think what would be really cool is if you can engineer cells inside of your body. And I can say why, right? Because that would be amazing, right? And I think and I don't think it's unrealistic. I think it's going to happen. It might take a while, but because just all the problems or all the manufacturing challenges that you face in actually extracting the cells out of a patient's body and then expanding them and then engineering them and then inputting them back again. If we could do everything in situ, or sort of like, that would be so much better and so much easier. Of course, I think we're still far. But this idea of doing modifications in specific cell types in your body, well, we're bound to it if we don't have a problem with global warming until then.
Yeah. Okay, let's see this one. Can protein folding start during protein synthesis and produce structures which wouldn't occur if the whole sequence was synthesized before folding started?
Yeah, that's a very insider question. Yes, so very, very insider. Yeah, so I think it's certainly something that we don't fully understand. I clearly simplified this idea that you go from sort of like an extended structure to something that it's collapsed. The way that this actually occurs in a cell, it's something that it's relatively hard to measure. And I think that's why it's also hard for us to know exactly what occurs during this process, because we don't have the experimental ways to measure it. And so it's hard to know. But if you like to have my guess, I would say that it probably matters. These intermediate structures that these polymers adapt, they're probably critical for the endpoint, I would say. But I think right now we basically don't have a lot of data to make larger conclusions out of that.
Okay, the next question. This science fiction technology you mentioned that takes cells out of the body and produces many and then returns them to the body, is it individual specific? Yes.
Good point.
Hold on. Sex, race, age specific? Can you import from one person to another? I mean, this is a whole day talk, basically.
Well, but I can actually answer that very quickly. So it's very personalized. It's very individual specific, absolutely. And I tell you, and the reason goes back to a lot of the things that I was telling you in my talk is because if I receive cells from somebody else, my immune system most likely will recognize that they're not mine, and it's just going to destroy them. So the way to go around that is, that's why this technology is not currently science fiction, but it's actually something that people can do, is because you're taking something that is self, and as such, and even though it's a bit modified or minimally modified outside of the body, when it goes back to the body, it's still seen as self. So your immune system is like, oh yeah, this is clear, it's no problem. Mm-hmm. Now, I told you that this was already current medicine, but I also told you that it had a problem in scalability. Exactly one of the problems of scalability is because you do not have a common platform, cellular platform that you can engineer and put in everybody in this room. That just sort of like doesn't go there, right? Because it's not going to work because of this self component to it.
Yeah. So I want to take a break a moment from the protein folding and immune system to get to stem cells. I know you haven't talked about them, but there is a question. Can you please tell us how stem cells fits into the picture of bioengineering and treatments?
So I am certainly not a specialist in stem cells, so I prefer not to say too much, but I will just say to the audience, for those of you that are not super familiar with stem cells, those are, in a way, sort of like these cells that still have the potential to differentiate in different kinds of cells. So you could imagine that, for any cell-based therapy, you would always start from a stem cell, and then if you would have the power to differentiate it to, let's say, a B cell or a T cell that you would like to then infuse into a patient, this would be the best way to do it. Again, I'm not a super specialist on how stem cell engineering is going, but I could imagine that if you would have this type of cell that you could then manipulate in a predictive way, of course, then that would be great. But I think that right now it's still very complicated.
Correct. Yeah. Okay, let's go back to, well, there is a question about mask wearing. So let's see. Are we more exposed to RSV as a result of obsessive mask wearing? What is the smartest masking policy? I know you're not,
that's definitely not my thing, but I'll tell you. So it does seem like, in fact, that has been the case from the reports that we see, that right now, because our immune system is not so constantly exposed to the circulating virus, suddenly when you put your mask down, you now, your immune system forgot that it saw this virus 3 years ago, so now you're not protected anymore. There's also this aspect that, again, because of RSV infections, I think we also had much more RSV infections before that we just didn't know about. But now, because people actually check what it is, you actually know there is an RSV infection. But, sometimes when you have this cold, it could very well be RSV, but we just didn't monitor that very well. But, there is something to be said about masking and about how you can clearly see that that's a way to avoid your immune system to be trained so that it can better respond to circulating viruses, right? So there's definitely sort of like this balance in between being sterile and in between sampling a bit of, the wild out there.
Yes. You compared modifying proteins to sculpting in a nano world. What kind of imaginative guesswork is involved? How do you teach this kind of creativity? Interesting question.
Yeah, well, so I think, above all, whenever you're trying to engineer proteins, most of the times you kind of have to have a dream. Mm-hmm. Okay, so like, well, what would I like my new protein to do, right? And if you don't have that dream, oftentimes it becomes pretty boring, okay? But then the way to teach it to people so that they can actually materialize their dreams is in a way, so like, you sit in front of your lab book or your computer and whatever, it's like, okay, well, so if I want a protein to do X, the next question is, which structure should this protein have? And to do that, right, at this point in your life, at least to do it the way we do it, you should already understand about chemistry, physics, protein structure, biochemistry, right? So in a way, you can't go there, at least currently, you can't go there just sort of like, oh, I just have this dream. You have to have this way. Okay, I have this dream, and now I realize it. How? And then once you have this hypothesis on how to realize it, this is how you start sort of like then engineering things, oftentimes in a computer, if you're doing research in our lab. And then you test them to see if in fact your hypothesis was right or wrong. So you can actually see this as basically a process of hypothesis generation, ultimately.
Okay, some big picture questions. Where do you see the future of protein engineering for society going?
Yeah. So I think vaccines is a clear application where we oftentimes, even if it's minor engineering, I think vaccines is clearly asking for it. Monoclonal antibodies, and maybe many of you don't know, but a lot of so there's many different types of drugs, and for instance, we've been seeing that monoclonal antibodies have been starting to be more and more used as drug modalities because of some of the good properties that they have. So that would be another application. I think also in the domain of biomaterials, where you can actually do smart materials that modulate according to the light or according to the environment. And then I think also, there's a domain which I find very interesting, which might be a little speculative, but it's really about our environmental concerns, right? Maybe part of the solution for all the environmental issues that we have in terms of carbon production, plastic degradation, might come from the domain of protein engineering. I am yet certain if one will not run into problems of scalability on how scalable you can make it.
Exactly.
But some of the solutions, at least in principle, might come from there.
Great. Going back to cell-based therapy, do you think the cell-based therapies will become a much more prevalent format for therapeutic intervention?
I guess, so right now they still have a lot of limitations, but this was true also for many different therapies 10 years ago that had a lot of limitations that were solved. So if you think about the potential that this type of therapies have, it's quite impressive, right? It's very impressive, right? Because suddenly you have a living therapy. So imagine that you have a living therapy that you put in your body that somehow adapts to the environment or to the stimuli that it has. Let's say that if it's night, it doesn't produce the drug because you're sleeping, so you don't need it. So you wake up, suddenly it starts producing the drug you need. That sounds like a pretty good idea to me, honestly, right? But again, of course, with all this power that these complex biological entities provide you, there's also a lot of risks, right? Because, we cannot really fully predict what these biological entities do in every situation. So there are possibilities for things to go wrong. And I think, safety is clearly a problem, right? We cannot just sort of like go out there and do everything we think it's cool. But we should definitely try it out. So I do think that if we have the possibility to scale this type of therapies better, there will be a range of diseases that right now we can't do much about it that will start becoming accessible.
Mm-hmm. Regarding your work for vaccine design, what do you think will be the trend for future vaccines?
Yeah, so I think related to vaccine design, I think one of the trends, and I think many of you are aware that the future is now and it's here, has been on the delivery systems. So delivery systems have been making huge improvements in what we can do with vaccines, right? And I hear the key word is mRNA, right? We could have never had the type of, how should I say, intervention that we've had in the past 2 years if we would have not been able to deliver vaccines the same way that we did with mRNA. So vaccine delivery is going to be certainly a very important aspect. The other aspect is how to engage. So right now, vaccines, sometimes they still don't do a great job into engage multiple arms of the immune system. So the idea here also in the delivery is how can we engage our immune system better so that these immune responses become more potent. So I think those are two aspects that are very important.
And then there is a kind of a personal question, which is, if you could discover any vaccine, which disease or illness would you target?
Yeah. HIV clearly is an important one, although HIV, currently people that are infected with HIV have very functional lives, and the therapies have come, or the medicine have come to a way that I don't think we could imagine. But I tell you something that I would do. Maybe I won't tell you an exact pathogen, but I tell you something I would do. If I could work on some technology, I would definitely work on a technology which could make vaccines available for everybody. That I would spend my time on, right? So like in this way, on how can we more easily bring this type of medicine to places where people cannot have it. Because again, we're here. Vaccines for us are a given, but for a very significant part of the world, that's not the case.
Yeah, and following up on that, does your work? Your work has applications that pharmaceutical company eventually will take on, but how does that transition happen?
Yeah, so that's actually, it's a constant dilemma on the life of a fundamental scientist that mostly wants to have fun, but however, there are some tools that can do things, right? So the translation from a basic discovery to an applied application, it's not a super easy business, and it's almost a science on its own, right? So just because you do something in a lab which looks good and it does everything you think it should be doing, I think until that actually becomes like a real drug or a real therapy or a real vaccine, many steps and oftentimes many years of research have to be done. And I really think that this can only really be made by very close collaborations between industry and academic labs, so that one can see if those basic research findings have a path to translation. Oftentimes, academic labs are not really well equipped to do that kind of research, and it's also not their mission oftentimes, because our mission is, or at least the mission in my lab, is to educate people and to make scientists and to teach people. But of course, we always pay attention to that, right? If there's something that has the potential, then let's see what it is. But I do think that the scope that we can do is limited, and as such, I think this crosstalk in between industry and academic labs is absolutely important to try to walk that path.
Mm-hmm. Actually, we are at time, so I'll keep this question.
You sure?
for. Yes, it was mine, so I can ask you later. So thank you so much, Bruno. Thank you for the fascinating talk and for answering all our questions. Thank you to the audience, both online and here in the room with me. There are plenty of programming at Radcliffe. You can find out more about future programming and video of past events at radcliffe.harvard.edu. Thank you for joining us today and have a great rest of the day. Bye-bye.
Thanks a lot.